Good afternoon. My name is John Wasleth, and I'm the Managing Director of Arrowhead Publishers and Conferences, the organizer of today's webinar. We produce both conferences and webinars for the life sciences industry, which include our annual Traumatic Brain Injury Conference, the Personalized Medicine Conference, Drug Repositioning Conference, and our annual Pain Therapeutics Summit, which is now entering its 8th year. This year's Pain Summits will be taking place in Boston on September 24th and 25th, and in San Francisco on November 5th and 6th. For more information about our events, please visit arrowheadpublishers.com. Today's webinar is scheduled to last for an hour and a half, with 1 hour for Dr. Neil Singla's presentation and 30 minutes for a question and answer period. Dr. Neil Singla is the founder and Chief Scientific Officer for Lotus Clinical Research, an analgesic research site and CRO located in Pasadena, California. Established in 2001, Lotus has 40 employees, a 40-bed phase 1 unit, the ability to run phase 2 and 3 analgesic programs on a site and or CRO level, full CRO capabilities, and most importantly, therapeutic expertise in pain. Dr. Singla currently chairs the Analgesic Clinical Trial Special Interest Group at both the American Pain Society and the International Association for the Study of Pain. The main focus of Dr. Singla's academic endeavors has been to analyze and understand how the inherent variability in analgesic clinical trials can be minimized. He has developed novel techniques for patient education designed to minimize variability, reduce placebo response, and increase effect size. Dr. Singla's affiliation with Lotus provides a unique opportunity not only to develop novel methodological techniques, but also implement and test them. Dr. Singla has frequent regulatory correspondence with the FDA's Analgesics Division on behalf of Lotus's pharmaceutical clients. Now I'd like to hand things over to Dr. Neil Singla.
Thanks so much for that introduction. I'm pleased to be here and excited to speak about our topic today, which is a summary and discussion of the new guidance document that was released by the Analgesics Division of the agency in February of this year. I had a chance to take a sneak peek at the attendee list prior to getting on the call, and I noted that there were a large number of old friends and collaborators. And to those who I know, hello, and to those who I don't, welcome, and I'm looking forward to getting to know you as time progresses. There's a lot of experienced drug developers on the call, and as such, I'm going to limit my remarks to 60 minutes and then be very interested to hear the comments and remarks of others, as I think we can learn a huge amount from the cumulative knowledge base of all the people on this call. With that being said, let me start on the first slide. I was interested to note, and it took me a few emails to conclusively determine, that the last guidance document was released 22 years ago in 1992. I Googled the term interesting event in 1992 to give some context to this time span and come up with something interesting. And I learned that the internet was first opened to commercial non-academic use in this year, which I thought was astounding. And as I searched, Internet Explorer gave me an error message, closed down, and forced me to restart my computer. So as much as things change, they also stay the same. Now, turning my attention to the guidance document, I want to first say that I think it's a huge step for the agency. It establishes a wealth of knowledge, and I would describe the guidance as a scaffolding upon which to build your development platform. That being said, it's a regulatory document, and as such, there's regulatory speak throughout, leaving only one thing absolutely certain after you read the entire thing, which is, well, the ultimate answer really depends. I don't know about you, but this is my first line of defense with my kids. No matter what the question, the answer is always, we'll see. Gives me time to regroup. But in defense of the agency on this point, I'd say that analgesic drug development is so complex that specific answers really do depend on the exact situation that the drug finds itself in. There's all kinds of ratios which need to be balanced, including risk-benefit, experimental control versus extrapolability, if that's a word. And there's a lot in this document that can help us with our regulatory interactions in that it basically speaks to the current mindset of the agency. Now, I previously described the guidance document as a scaffolding, and I want to expand on that concept. My feeling is that each regulatory interaction, whether it be in person or in writing, is critical. Both the agency and the sponsor have only a certain amount of time or resources and really mind share. So during each touch point, something critical should be learned. As much as possible, you want to avoid debating issues that the agency has already taken a certain stance on. And they've laid out many of these issues, luckily, in the guidance document. And as logical as this seems, it's really a common pitfall. But let's remember that the guidance document has only been out for a couple of months. So sponsors and consultants alike really couldn't previously be sure what was dogma and what was more fungible. And now we've taken a step towards knowing, which is a big help for all parties. So what should you focus on in your meeting? Focus on what really matters. And some of these issues are as follows. Big ticket issues. What clinical populations, in other words, disease conditions, are to be studied and are appropriate for this investigational product? And should those studies succeed, what indication would be granted? This is an important question to ask because you might be surprised at the answer, and that answer has large repercussions. What specifically should be the choice of the primary endpoint, and how should it be measured? With what scale? What key secondary endpoints should be chosen, and if they're positive, and if you measure them in way X, Y, or Z, which by the way is another question, those endpoints are positive, would those positive findings be translated into the label or not? How many total safety exposures will be required? And this one sounds potentially odd, but does the agency have any what I call bee in their bonnet issues about your drug? You'd like to know that early. I'll convey a little bit of a story. I was at the agency 2 years ago at an end of phase 2 meeting where we were planning our phase 3 studies, and approximately 20 minutes in, the deputy director at the time said, you know, I have concerns about the drug in that there are a few safety concerns that are going to be very difficult to overcome just because of powering issues. Your drug does have efficacy and merit, but it's going to be a difficult path forward. Now, you're welcome to put a plan together, and I'll definitely review it, but essentially indicated that the stance was going to be unfavorable. Now, as depressing as that is, it's actually an extremely important comment, and you'd like to get to it rather than move on for a year and come back after spending a lot of time and money only to get a negative response. And that is really the highest purpose of these meetings. Now, the specific topics that I plan to discuss are the regulatory pathways that one can choose and the labeling implications based on the pathway that is chosen. The guidance document actually spends a significant amount of time describing this, and it's important. Then I'm going to compare and contrast the 8 most common analgesic models. 4 of them are in acute pain, 4 of them are in chronic pain. I want to cover techniques to maximize assay sensitivity, which is a huge problem in analgesic trials. Secondary endpoints, there are several regulatory hurdles, and secondary endpoints have really important commercial implications. I want to discuss that. Rescue medication, there's ways to minimize its experimental impact. How do you deal with dropouts? How do you impute data? A large amount of time is spent talking and thinking about this. A little bit about safety studies. Then we're going to cover phase 1 considerations, 2 main considerations, which are safety considerations and stopping criteria, and then a general overview of phase 2 programs. So clearly, we have a lot of ground to cover. The first choice developers face is what disease population should be studied? And the next question is, should those studies be successful, what is the likely indication that would be granted? The FDA has indicated that the label will reflect only clinical populations that have been studied. Clinical programs should support the logical clinical intended use of the drug, and relatively narrow claims should be avoided and will be granted only when appropriate. An example is an intra-articular injection into the knee of a new analgesic. If you did 2 osteoarthritis studies for a drug like that, then you probably could get an indication for the treatment of osteoarthritis of the knees of such and such severity, etc. But if, for example, you were developing a transdermal sufentanil product, which is a narcotic, obviously, and you did 2 osteoarthritis studies and presented them to the agency, that probably would not be appropriate because obviously if it is positive in osteoarthritis and the drug gets on the market, there's going to be off-label use. And the agency wants to discourage this type of labeling and behavior. They want to get an idea of how the drug will actually be used and then grants an approval based on a variety of studies that mimic this actual use. In this slide, I'm presenting a flow chart and categorizing pain conditions in the way that the agency has categorized them in the guidance document. From a regulatory standpoint, this flow sheet has significant impact because it establishes the basis upon which indications are granted. Now here in the green boxes, you see actual disease states that fall into each category. These clinical populations represent study model choices, and there's some good news, bad news here, which is that most of these models are so difficult to recruit and medically complex, in other words, experimentally confounded, that they really don't lend themselves to successful and practical clinical trials. Now, by the way, that's the bad news. Here in the next slide, we see a little bit of good news in that if you delete the models that are extremely difficult, you're left with essentially 8 models that have been tried and true for drug developers over the years. 4 models in acute pain and 4 models in chronic pain. And these are the models I'm going to focus on comparing and contrasting in the next 2 slides. I know that previous slide was somewhat busy, and you probably want to know which models I was speaking about, and here they are. Here are the 8 models that I was previously discussing. For chronic pain, they are OA, low back pain, DPN, and PHN. And for acute pain, they are third molar extraction, bunionectomy, total joint replacement, meaning knee or hip replacement, and visceral surgery, usually hysterectomy, hernia, abdominoplasty, that type of surgery. I've selected these 8 models based on the following premise. We as scientists are standing on the shoulders of giants. We know what we know about certain models because they have been, the methodology of those models have been studied over time. So if we pick new models in which there has not been a lot of clinical study, then we are experimenting on the model and not experimenting on the drug. If you have a drug that doesn't fall into one of these models nicely, then you have to choose another model, but that is a difficult position to be in. So if you're not in that position, then choose something tried and true. Okay, and one more sweeping generality, which I'm sure will generate debate, but I'm going to put out there, is that acute pain is much less risky as far as it has a higher assay sensitivity. It has a faster recruitment time and a lower cost than chronic pain. At the same time, chronic pain has more commercial importance because more people suffer from chronic pain, and therefore there are more dollars to be had in that market. And although acute pain is undertreated and it's very important to find new therapies for acute pain, it is extremely important. It's probably more important from a humanitarian aspect to solve some of these very difficult problems that we have in chronic pain. But again, both problems are important. These are just generalities. Let me turn my attention to the actual table. The columns are categories relevant to developers, and let's start with the first one, which is assay sensitivity. Assay sensitivity is the signal-to-noise ratio of the study, and one can think of it as the risk that a negative finding will occur even if the IP is effective. The second column is recruitment time, which is a function of both the prevalence of the disease and the mechanisms that research centers use to recruit those subjects. And the third column is of critical importance, of course, which is cost. Cost is a function of time, but also a function of the logistical difficulty in recruiting and handling patients. And I'm basing my estimation on my experience costing studies at both a site and CRO level, but I'd be interested in hearing what others think in the Q&A session. Commercial importance is also listed, and I think I've already discussed that chronic pain has a larger market, and even though acute pain is relatively undertreated and we have a need for better acute pain drugs for sure, the market is still smaller. Now you'll note one very interesting point, which is that there is no green line that extends all the way across any model, meaning that that model would be both easy to do and commercially very important. Whereas acute pain is easier, it's not as commercially viable. Chronic pain is harder, but more commercially important. And isn't that life? And this is why a lot of sponsors will choose to move forward first within a strategy in which they get a drug approved for acute pain and then turn their attention to chronic pain. Of course, that strategy only works if you have a drug that's efficacious in both acute and chronic pain conditions, which is not always the case. So now let's briefly discuss the evidence behind these rows and columns. Relevant to the acute pain sections, our group published this meta-analysis and review in IASP's journal last month. Essentially, the paper rank orders acute pain models for assay sensitivity and recruitment time. It doesn't discuss cost. These estimations were made based on my experience and the experience of my team. The commercial and humanitarian columns are opinion. I'd be interested to hear what others think. Regarding the assay sensitivity and recruitment time of chronic pain models, these are also based on research that our group has done but not yet published. So on this next slide, we can see that when we rank order effect size according to the research that I just mentioned, here's what we have across all of the 8 models. The reason I'm pulling assay sensitivity out as an entirely separate slide is because it's probably the most important column. Why? Because it affects both risk and cost at the same time. You can see here that as effect size goes up, take dental in green as the example, you need far fewer patients per arm to get a positive signal. And as effect size goes down, take DPN with a 0.3 on average, you need to be in the mid-300s as far as N, which has obvious cost and time implications. Taking the 8 best studied and most viable options that I mentioned, and then superimposing them on the agency's algorithm, one can begin to pick and choose their pathway in a more logical fashion. The agency spends a significant amount of time in the document indicating what label will be granted based on how many studies are done in each of these pain categories. And rather than review it in depth here, I'll let you refer to the guidance, but it's clearly spelled out with, of course, clear in quotation marks based on the "it depends" backdrop. But it's definitely worth checking out. Okay, phew, that was a mouthful. Now we're going to regroup and change topics to discuss mechanisms to improve assay sensitivity. In other words, increase and maximize those effect size numbers that I laid out in the previous slides. What's important to note is that the effect size numbers populated in those previous tables are based on studies that are currently in the literature. They're not unchangeable. So as we improve our methodology, there's no reason to believe that we can't make those numbers better. So yes, it is true that the effect size is a property of the drug being studied. In other words, the tip of the triangle here. But effect size or assay sensitivity or signal-to-noise ratio is also impacted by 2 other factors that we do have more control over, which are a well-written study protocol here on the bottom left and optimized study conduct on the bottom right. You generally need all 3 to have a positive study. In the next slides, we'll mathematically explore why that's the case and then put some solutions and evidence on the table. The top panel here is simply the mathematical formula for standardized effect size, which is drug effect minus placebo effect divided by the pooled standard deviation or variability that was observed. In simpler terms, signal-to-noise ratio of the investigation. Let's turn our attention to the bottom panel, in which all patients more or less, as you can see on the y-axis, enter the study with a pain intensity of 5. And then as they continue in the study, the x-axis is time, their pain intensity changes over time. The red line is the patients in the study who received placebo, and we can see their pain gets slightly better. The black line is patients who receive the investigational product, and the green area between these two lines is a graphical representation of the standardized effect size. So we want to maximize this green area, and how do we do that? We do it in 3 ways. Number 1, we attempt to maximize drug effect by giving the appropriate patient population, in other words, a population that is going to respond to the drug, we give them the drug at the appropriate time course in their disease state. And this is determined and optimized mainly by good protocol writing. Then we attempt to reduce the variability in between patients, and we can do that by using as few centers as possible, standardizing questionnaires and educational processes so that all subjects are answering the same question and you get less random artifact. And lastly, we want to reduce the placebo response in red here, and we do that by educating patients and educating study coordinators and study staff to reduce positive bias, control expectations, and stay objective. On this subject of placebo response reduction and control of subject expectation, we have both a subject and an investigator training series that we deploy in our analgesic trials. I'll briefly give you an example of the patient education series, and then later in the program, I'll give a few examples of the investigator education series. But since we have so many topics to cover, I can't extensively describe the system. But briefly, the patient series is approximately 14 minutes. We have the patients educated when they screen and also prior to randomization. The series discusses placebo response, subject expectation, attempts to standardize the 0 to 10 pain scale, and then discusses the primary endpoint for whatever study is being conducted. So in other words, if it's an OA study and using the WOMAC, then we have a series on the WOMAC. If it is an NPRS, then we spend a lot of time discussing NPRS, et cetera, et cetera. And here's one of the chapters, which discusses therapeutic clinical relationships that doctors and patients have and contrasts that to the research relationship. Now, because the webinar doesn't stream video well, we elected simply to include the audio and a still shot of the video. In reality, when we stream this over the web to study participants or distribute it, it's very easy to distribute, but for some odd reason, it's not well done over webinars. And so just imagine that these people are talking in a video, even though you're just going to hear the audio and see a still shot. When you are in a treatment relationship with your healthcare providers, the goal of both parties is to do whatever it takes to get you better. It's a partnership that is therapeutically aligned. In research, while the study team is there to care for you and keep you safe, the main goal is to conduct the study in the most objective way possible. For example, if you were a patient on a typical hospital floor, the nurses would check in on you frequently and ask if you needed any medication for any discomfort you may be experiencing. They're trying to help you in any way that they can. If you needed help, they certainly would not give you a placebo. As a study subject, the research team will monitor and care for you, but they have two goals. The most important, without question, is that you stay safe. But the main objective of the study is to get the data in an accurate way. We have highly trained physicians, nurses, and research staff that will be working to meet the objectives for the study, as well as providing you with excellent care. Okay, so I mentioned some other chapters. The things I forgot are understanding when to use rescue medication. We talk to subjects about that. We talk about when they should consider dropping out and when they should consider staying in and what are their options regarding that if they're thinking about dropping out. And an important part of our subject education process is that after the subjects watch the video, we have a post-test that is administered by the study coordinator. And what that allows us to do is really identify cognitive gaps that the patient may have even after watching the material. The test sets up vignettes for the patient. So, for example, it will say a patient, Jane, came into a research study, came in with a baseline pain of 8, really liked her study coordinator, really liked her investigator. The pain went from 8 to a 7, back up to an 8. She took a lot of rescue medication. Now she comes in and has to give her global assessment. Should that global assessment be excellent? Should it be poor? Should it be fair? Should it be good? And you get a lot of subjects saying it should be excellent because the doctor was excellent and the nurse was excellent. Well, no, in fact, it shouldn't be excellent. The drug in the vignette clearly didn't work. And we want patients to cognitively understand that. They are ranking the drug, not ranking the experience of the research staff or the amount that they like and appreciate the research staff like you do when you leave a hotel or something like that. The investigator and staff training series is also of critical importance. We deploy this at the investigator meetings and then subsequently over the web for people who can't attend the investigator meetings or investigators who change and whatnot. And it goes over a variety of topics, but most of them have to do with how you present the protocol to your patients, etc. So the series has nothing to do with the actual protocol. It has to do with the gray areas that are prevalent and need to be understood, the gray areas in conducting analgesic research. For example, don't say to your patient, hey, I'm really excited about this drug. The phase 2 results were really promising. You don't have to participate, but if you're randomized to active, then I think, you know, you might have a good effect. You want to stay away from any comments like that. And of course, the series is a 1-hour series, so it goes into lots of different topics, including dropouts, etc. But that's basically what it's about. And what evidence do we have to support that this patient and investigator education works? We are using the education series right now in a prospective randomized analgesic trial in which one group of patients is educated and one group of patients is not educated. That's one thing we're doing. Results still pending. The results displayed in this slide were garnered in the following way. We at Lotus participate in many multi-center clinical trials, which means that all centers are performing the same protocol on similar subset of patients because they all have to meet pre-specified inclusion-exclusion criteria. So we're all doing the same study with the same drug on the same patients. And in these investigations, we pull out centers that use and have placebo response tools versus those that don't, and interrogate the difference, if there is any, in the effect size. And in the studies that we have examined, we have seen that there are very positive differences as displayed here. Okay, now switching to our third topic, which is secondary endpoints. First of all, let me say that secondary endpoints are commercially incredibly important. A claim in the label goes a long way to selling the drug. It's a precarious position if you think about it, in that in analgesic studies, it's extremely hard to hit your primary endpoint, but commercially, in many situations, you're judged by your secondaries. If your secondary is positive but doesn't make it into the label, this is a big commercial difference for the sponsor pharmaceutical company. And I'm going to share some experience I have because I'm a medical speaker for 2 or 3 drugs that I've done research on over the years and used in my clinic. As a speaker, you have to go through extensive training. The training, which I can summarize, is basically stay on label, stay on label. If someone in the audience asks you something off label, clearly identify that it's off label and then switch back quickly onto the label. So, and field representatives are even put under greater scrutiny. You're supposed to have some freedom as a physician who's prescribing the drug to discuss the situation with other physicians in the audience. Field reps don't have that freedom. So the point is, if it's not in the label, you can't and shouldn't market with that claim, and that really limits you commercially. So secondaries have importance, and we'll move to the next slide, which is how do you get these secondaries into the label? Now, earlier, probably the first or second slide in the talk, I discussed the best and highest purpose of any agency interaction or meeting. And I specifically highlighted things that should be accomplished during the interactions or meetings. What I failed to point out, and am going to try to point out as the presentation continues, is things that should be avoided and that we should try not to waste time on during our regulatory interactions, because the agency has tried to make clear their stance on certain subjects like secondaries. So in the guidance document, they state that if you want a secondary to be included, or even a primary, but they talk about secondaries as well, first of all, you have to measure that secondary using a validated PRO. Now that sounds simple, but for many domains of interest, there is no validated PRO. And when you come to the agency with your best try, and you think, well, okay, there's nothing out there, and we want to measure this domain, and we'd like it in the label because our drug really does X, they are going to say, this is very interesting. We actually think that your drug might do this, but in order to get it in the label, you're going to have to use a validated PRO. And if you would like, we would encourage you to go out and validate this questionnaire that you want to use as a PRO. Now, they're saying that because they need more validated PROs, and there are companies once in a while that are willing to do that. But in general, that's an extremely arduous process. It takes a lot of time, and really you can't delay your program long enough in order to get a validated PRO to measure that. So you really end up relying on whatever is currently already out there. So the quick point is, don't waste your time making this argument. You'll go back and forth a lot. I've done that myself. I suggested that we use the ORSDS, the opioid-related symptom distress scale, to quantify the benefit of an IP I was studying in reducing opioid-related symptoms. I had seen that scale in several academic papers and in several registration studies, so I assumed it was validated, but I was wrong. It hadn't gone through the sealed team at the agency and really wasted the time of my client for 2 or 3 months going back and forth making this determination, so I learned that lesson the hard way. Other issues about secondary endpoints is that they must be pre-specified. You can't go back afterwards and go, oh, you know what, we had less nausea, let's ask to put that into the label based on the adverse event data, even though we didn't pre-specify that endpoint. That's not going to work. Secondaries have to address multiplicity. And I know there's many people that completely understand this, but for those who don't, essentially, if you claim or try to look for 20 secondaries at a p-value of 0.05, you have a 1/20th chance randomly to obtain any claim. And so the agency doesn't want to see that you listed 20 secondaries and you hit 2 of them, because mathematically that would be logical and really doesn't mean that those secondaries are in fact truly indicative of something the drug is doing. It might just be random chance. How do you address multiplicity? Well, there's two major ways. I'm not a statistician. I'm sure there's many statisticians on the phone. But in general, number one, you can rank order them, meaning that once you list 20 secondaries in a row, if you hit secondary number 1, then you move on to secondary number 2, then you move on to 3, et cetera, et cetera. Once you miss one of the secondaries, you don't test for significance for any of the secondaries below it. That's one way. Or you can take a statistical penalty and adjust for multiplicity in a way that's more complicated than I really am qualified to discuss, but there are ways to do it. The next point is that the secondary that you choose should be clinically relevant, and I'll give an example of a situation that I was just in. We had a drug that we were exploring, and we thought it was going to reduce the subject's opioid burden. This was a different drug. Opioid reduction is clearly an important secondary for all analgesic trials. We'll get to that in a moment. But anyway, we thought it would reduce the patient's subject's analgesic burden, opioid burden, and as such, patients would have less respiratory depression, a sign or downstream symptom of which is better oxygen saturation. So we suggested oxygen saturation as a key secondary, and the agency replied by saying, you can collect oxygen saturation differences, and even if it's statistically significant, we are not going to give you a label claim for reduction of opioid symptom distress or for less respiratory depression unless those differences are not only statistically significant, but also clinically important. And they take that view, which is a logical view, for any secondary. So again, this is something to accept and not to debate during your meetings and get to other things which are more important, and you have a chance of making a positive influence for your program. In other words, the scaffolding is already set. Don't waste your time trying to pull it down. The last point I'll make on this is that your secondary should represent findings that are not directly a result of change in pain intensity, and the agency specifically calls that out in the guidance document. The example they provide is this. If you have really bad OA, that means you can't sleep because all night you're in pain. Now you give this miracle drug for OA, it cures the pain of OA, and patients sleep better. They don't want to provide you a secondary of improved sleep, even if you have a validated PRO to measure it, because the improved sleep is simply secondary to the fact that the patients have less pain and not because the drug actually helps you sleep. So I made that point as well. Okay, the last point I'll make on secondary endpoints is that planning for success with secondaries starts when you are in proof of concept. And this is because we've enumerated previously the difficulties with secondary endpoints, namely that you have to find a validated PRO, you have to figure out a way to ensure that it will be in your pivotal studies clinically significant, and statistically you have to power it appropriately, which is difficult to do. Now, because you have an investigational product, most likely you won't know which secondary could potentially meet all of these criteria. And so in phase 2, you explore a range of secondaries. And it's not for the purpose of a label claim, obviously. It's for the purpose of understanding and powering your phase 3 program so that you have a reasonable chance of achieving that important secondary, getting it into the label, which is commercially very important for most drugs. Again, questions that need to be answered in POC: which secondaries, what type of scale, with what cutoff, and how much power are you going to have, and therefore how do you need to populate your pivotal programs in order to achieve your goals. Okay, now let's turn our attention to the use of rescue medication. I like to introduce this topic by speaking about rescue in two ways. There is an experimental concept with rescue and an ethical concept, and the two are often conflated. By separating them and clearly understanding them, we can learn and think about how to deal with rescue properly. First of all, from an experimental standpoint, rescue is a confounder. The impact group did a nice meta-analysis of this, and the general idea is that the more rescue you give, the better it works, the less it is regulated, the more the pain intensity between the placebo and active treatment lines will come together. In other words, a degradation in signal when you use more and more rescue. So why do we use rescue? That's the ethical concept, because we have patients and subjects in placebo-controlled clinical trials, and we have to and should use it. So if this is true, then how do we mitigate the effects of rescue on assay sensitivity? And the way we do it is by pre-specifying the following issues regarding the administration of rescue drug. First of all, what kind of rescue are we going to be allowed to give? It needs to be very restrictive and well thought out in the protocol and well delineated. What pain intensity should the subjects have prior to getting the rescue? Now, you can't mandate that because patients can take rescue medication whenever they want. That's part of ethical conduct of analgesic research. But you can suggest it. You can educate patients toward it. You can educate staff toward it. You can monitor it throughout the conduct of the trial and re-educate staff if appropriate. How much rescue and at what time points should the subject take it? My view, after experiencing a significant number of patient interactions, is that subjects, for the most part, really do want to follow the protocol and understand it. We sometimes fail to explain it to them. So making sure they know, this is when I want you to take the rescue, and don't take it before bed just because you don't want to get up in the middle of the night and you're worried that you might have pain. Take it at an appropriate time. This is an important discussion. You want to pre-specify the timing of the rescue medication in regards to when the scheduled assessment is. So you might say, well, we discourage rescue before 30 minutes before a scheduled assessment, depending on the study and the clinical situation, et cetera. And another thing you want to do is collect pre-rescue pain assessments in most clinical situations. So right before the patient takes rescue, they tell you how much pain they're having. And why do you do that? Because you use it for different data imputation strategies that we'll discuss later in the talk. And lastly, the agency has made it very clear that they want the amount of rescue medication that is taken in a study between both arms to be captured, compared, and quantified as a key secondary endpoint. Now, I alluded to our education series, and what I'm going to do is show a quick, video and audio snippet of our investigator education series that has to do with the use of rescue. This is an interaction between doctor and subject that we follow from when the subject gets consented to various treatment visits. And at this visit that you're seeing kind of the middle of our education series. A subject comes in and the coordinator is discussing how and when he should use rescue. So I'll let you listen to this. And again, these are stills only based on the technical limitations of the webinar streaming video as we previously discussed.
When should I take the rescue medication?
Well, the rescue's there to help you. So take it early and often. Remember, you could be getting placebo. We wouldn't want the pain to get ahead of you.
Okay, now obviously, this is not the way we want the coordinator to discuss rescue with the patient. We go on to talk about why that is and educate those coordinators and investigators as to what is the effect of rescue on clinical trial outcomes. Because I've been at many investigator meetings where investigators tell me, these are very experienced investigators, they say, well, listen, the amount of rescue is captured as a key secondary, and therefore it doesn't affect the primary. And that's just not true. And I think I can see how very educated people could think that and understand that, but this is an issue that's often misunderstood. Having people understand the statistical underpinnings and then showing not only that video where the coordinator is talking to a subject, but also a subsequent video in which the coordinator discusses it with the subject in a more formal or more favorable way. This is how we get to the point. And here now I present the more favorable or optimized way in which that same study coordinator should talk to a patient when they inquire about rescue.
So when do I take the rescue medication?
Now, at any time during the study, if you feel that your pain is not being adequately controlled by the study medication, you will have the option of taking the rescue medication, oxycodone. You should only take the rescue if you feel as though you need something to control your pain. You're allowed to take it no more frequently than every 6 hours, and it should not be taken preventatively, used only as treatment.
Okay, next topic is patient dropouts. Now here with patient dropouts, an ounce of prevention is worth a pound of cure. There are statistical ways to deal with dropouts, and this becomes a big topic of discussion during regulatory meetings. But to start at the beginning, if there's not enough data to make accurate conclusions because the dropout rate is too high, then you're going to have a serious problem that no statistical manipulation can deal with. So the best way to deal with dropouts is to deal with them proactively and try to prevent them. How can you prevent dropouts? The first is to allow appropriate rescue medication and enough rescue medication, but this is a double-edged sword because we just got finished talking about how rescue medication is a confounder. So no perfect answer for this, but if rescue is inadequate, then you're going to increase the number of dropouts. That's frustrating, isn't it? On the one hand, you have a slide telling you to do X, and on the next slide telling you to do the opposite of X. A second way is dose titration, and the guidance document actually spends a lot of time talking about that dose titration, especially for opioid-related studies, that patients need to build up on opioid doses. We don't just, in clinical practice, put patients on huge doses of opioids. We start at dose X and we escalate, we titrate. So clinical trials can incorporate these type of titration schemes either before or after randomization based on appropriate statistical care and interpretation of the results. And the other way we can prevent dropouts is through, again, educating both subjects and investigators. The subject education chapter spends time talking to the subject about the following fact, that number one, they can drop out of the study anytime they want, but if they're considering dropping out, there may be alternatives that they should feel free to explore with their doctor. For example, they should make sure they're taking the rescue medication appropriately. I've had many patients that say they want to drop out because they don't have transportation. Maybe that can be provided. If they think they need to drop out because they're going to miss the next visit, but could then subsequently make it to visits in the future, which are actually more important to the primary endpoint, especially in a normal 12-week study where you're comparing week 1 to week 12. If they miss week 7, you would much rather have them stay in the study and give you your primary endpoint data at week 12. This may be something that the subject doesn't know. So basically what we say in the subject education series is that dropout conversation is a conversation you can have with your doctor. Before you get in the study, think about that. Think about whether you have the time, energy, and resources to be in this study. And this is how we try to reduce dropouts on the patient side. Now, on the investigator side, we also have a video education series. And I'm going to show you a snippet of this as well, where a patient comes in and talks to his doctor about potentially dropping out of the study. You'll hear the doctor's response, and then we'll make some comments afterwards.
I've been in this study for a couple months now, and I don't seem to be getting any better. I think I'm taking all the drugs correctly, but I guess I'm not sure because if I was, I suppose I would be feeling well. Should I just drop out of the study?
Well, since you aren't getting any better, I think you should stop participating in the study. And I'll give you something stronger for your pain.
So we make first strongly the point that if a patient wants to drop out of the study, it's their right to drop out at any time without question, period. But if you notice in this scenario, the patient asked the doctor, well, I'm not getting better. What do you think I should do? Should I drop out? And without really knowing all of the information, the doctor just says, yeah, go ahead. I think you should drop out. And as indicated here on the next slide, we want the doctor to see this dropout conversation as just that, a conversation, and talk to the patient about rescue drugs, pain severity, any safety concerns, and their attitude towards completing the study, and then make an informed decision about whether or not the patient should drop out. Okay, moving along, we've covered a lot of ground. We're going to switch to another topic, which is data imputation. And basically what data imputation is a statistical methodology to deal with missing or confounded data. In the case of patient dropouts or missed assessments, it's missing data. And in the case of the situation where a patient takes rescue right before a scheduled pain assessment, that pain assessment is contaminated by the rescue. And certain data imputation strategies try to deal with that. We're going to talk about rescue first by looking at a graph and doing a little primer on what different data imputation methodologies there are. And then the next slide will be on the regulatory strategy to deal with data imputation, because this is a major point of discussion in FDA meetings. I think because their position on the issue has been evolving, and now it seems to be somewhat stabilized. And so I'm going to present to you what has been laid out in the guidance document and what I have seen the agency agree to, with the hope that if your program does in fact fall into the category of programs that could use this strategy, that you can essentially avoid wasting a lot of time in your regulatory interactions talking about data imputation. But first, let's do a primer, and then we'll go over that strategy. Okay, for the primer, let me set up this graph first. You have pain intensity on the y-axis, let's say an NPRS scale, and then time on the x-axis. This time could be days, weeks, months, whatever. In this case, we're going to make it hours, just because that's what we decided to do. And on the first click here, you see that patients enter the study with a baseline pain score of about 6. That, as I said, establishes their baseline, which is what that dotted line is across the graph. Then they have these blue stars, which are their scheduled pain assessments throughout the duration of the trial. In this case, it's a 10-hour trial, but again, could be 10 weeks, 10 months, whatever. Now, in this example, there's only one line, and in a real clinical trial, there would be at least 2 lines, one active, one placebo, and you look at the difference between those lines. But for the purposes of data imputation, let's just assume that we're talking about one group of patients. This is the placebo group, let's just say. Okay, moving on with the next click, we see that at hour 2, this patient takes rescue medication. And in this hypothetical protocol, there's 2 types of rescue medication you can take. One of them is acetaminophen. And the technique I'm going to demonstrate here is called windowed last observation carry forward. So this patient at 2 hours took acetaminophen. And the next click here, you can see this red line is carried over for 2 hours. So you get an assessment of the patient's pain severity prior to the administration of acetaminophen. Now you take that pain intensity and carry it over for the duration of action approximated of acetaminophen. And even though your coordinators collect the scheduled pain assessments in red, you don't statistically count them. You drop them. And that's what windowed LOCF is. Now in the next click, this subject took another dose of rescue medication with a different imputation window, in this case oxycodone, and a 2.5-hour imputation window. And the next click, you can see that the score of 3 is carried over, but now it's carried over for 2.5 hours, so it's carried over farther. So the amount that it's carried over depends on the type of rescue that is taken. So we've just discussed windowed LOCF and data imputation strategies as they relate to dealing with confounded efficacy measurements. And now we're going to talk about data imputation as it pertains to missing data as opposed to confounded data. So with this next click, we see that the red circle indicates, it's a little bit behind that blue star, indicates that there's an early termination due to lack of efficacy. So the patient was having pain and they terminated. Now in this situation, the pain is 2, and we would hope that patients don't terminate with a 2, but just for the point of demonstration, this patient terminates because of a 2, and the agency has said, okay, if patients terminate due to lack of efficacy, then LOCF, or last observation carried forward, as indicated in this next click, is a fair data imputation strategy to take, because whatever medication the patient was on is not working well for them, and therefore you can impute that data forward for a reasonable period of time. One would assume that patients in the placebo arm will rescue more due to obvious lack of efficacy, and therefore longer imputation windows tend to dampen the placebo response. In other words, make it statistically appear as if placebo patients had less pain relief. But regardless, the imputation windows have to be based on something logical. I just wanted to point out that phenomenon. Moving on now with the next click, what happens if a patient terminates due to lack of tolerability? That's indicated here in the green circle. So patient had a pain of 2, but think of it this way. This patient got good pain relief. They went from a 6 to a 2, but now they're having nausea that's so bad that they have to terminate out of the study. Now, what the agency does not want is for sponsors to use LOCF in this situation, because if they do, they get credit for these low pain scores at a time when a subject couldn't tolerate the medication. So with the next click here, you see what they want to be done, which is, in general, they want you to use BOCF, or baseline observation carried forward, for tolerability dropouts. So take that red line, go back to the baseline, so that from the point of dropout, you really get no more credit for a low pain score, because this is a patient that couldn't tolerate the medication, and therefore one doesn't deserve credit for that. And with the next click, you get the area under the curve for this patient with all of the different imputation methods employed. If you have done this many, many times, then this graph is very obvious to you. If you have not, then it's very difficult to understand. And getting a biostatistician who doesn't do this to understand it, it can be a very arduous process. So from a CRO standpoint, what's very important is that, number one, you have a biostatistician who knows how to do pain, understands the methodology and the imputation techniques. Number two, your CRF data capture gets pre-rescue VASs, captures the type of rescue medication that was used so that the imputation windows can be appropriately calculated. And when there are dropouts, there is a thorough questioning of the patient as to why they dropped out. Was it an efficacy dropout? Was it a tolerability dropout? The CRF collects this information along with a pre-dropout pain score, and all of this information is fed nicely on the data export from the EDC system to the biostatistician. Now here is the data imputation regulatory strategy that I promised. All specific programs need to have their own tailored strategy, but this strategy that I'm presenting here will work in 80% of the cases. First of all, windowed LOCF to decontaminate efficacy measurements confounded by rescue, as was discussed previously, LOCF for efficacy dropouts, baseline observation carried forward, or BOCF for tolerability dropouts, and then be sure to have a pre-specified sensitivity analysis. Specifically, the agency always talks about and is very favorably inclined to the National Academy of Sciences report, the prevention of treatment of missing data in clinical trials. It's publicly available. Just Google it if you don't already know everything about it. And there are multiple ways to perform sensitivity analyses, but one way is to use the mixed effect model for repeated measures, MMRM. And this is kind of at this point, a buzzword inside the agency. And if you mention it and you say, not only are we going to pre-specify all of these endpoints, but we're also going to do a sensitivity analysis, and we're going to deal with dropouts in this way, we're going to deal with rescue medication in this way, and then you mention the word MMRM, that's going to bring a smile to their faces in general. I mean, it has to be appropriate, and I'm saying that most of the time it will be. And then you're going to be able to move on to more important considerations. Moving on to the next slide, I wanted to take a minute to talk about safety exposures. And the key features of a safety study, which are that safety studies should mimic clinical practice. And by their nature, if you mimic clinical practice and don't control certain issues, efficacy endpoints are confounded. And the randomization generally does not need to be one-to-one, which means that you can pick up safety exposures more quickly in a safety study than you do in an efficacy study. Safety studies are generally easier to perform because the inclusion criteria are more liberal, and that makes recruitment faster. And the efficacy measurements, since it's a safety study, are generally less rigorous. Okay, now I want to get to the point that is really important in this slide, which is the bottom, and it says common pitfalls. Because the issues that I just discussed about a safety study are well known. But what goes wrong when this actually happens is that often safety studies become efficacy studies, and that's what makes things much more difficult, and I'll give an example. So you're at the end of your clinical program. You are interested in hitting a potential secondary endpoint, which you didn't achieve in your pivotals, or you're worried that you might not achieve. And now you're designing the safety study because you need a certain number of safety exposures, and you begin with a 3-to-1 randomization or some such thing. And you think, well, if we're going to spend all this time, money, and effort to get this study done, maybe if we retool it just a little bit, we can hit that secondary efficacy endpoint that we want, and we can get it into the label. Now, if you do that and you do hit your secondary, then yeah, maybe you could get it into the label. But what generally ends up happening when you start refining a study so that it looks more like an efficacy study is that all of a sudden it's more difficult to enroll, it is more rigorous to perform, it's not as quick, the randomization is 1-to-1, so you need more patients in order to get your safety exposures. In other words, you take a study that was previously supposed to be easy, faster, and cheaper, namely a safety study, and you've created a more difficult efficacy study. So this is something to think about a priori, to not move down and think, well, we'll pick up our exposures by just doing more efficacy studies, because that's really an inefficient way to do it. Regarding early phase studies, I'll make 2 points. First, the agency specifies in the briefing document that we strongly recommend that sponsors include in the protocol detailed information for managing adverse events, along with documentation, and this is the key part, of the immediate availability of staff capable of managing emergencies, meaning trained in airway management. In general, reliance on transport to an emergency room as the primary support for emergency events may not be appropriate. Now, this is a pretty strong statement and in many situations hard to live up to, because if you look at the idea of trained in airway management, I don't think that means research coordinators or even medical doctors who have been trained in ACLS or may have intubated patients during their residency. Trained in airway management means personnel that routinely manage airways. This is anesthesia parlance. Being an anesthesiologist, we talk about trained in airway management all the time. Now, to have someone like that available 24/7 is difficult. And for every early phase study, probably not appropriate. If you're doing a phase 1 study on a new NSAID, it's very unlikely that patients are going to have respiratory depression. But the agency did put this in the document, and I think there is some wiggle room. But if you're dealing with a new narcotic, etc., making sure that the research center you're looking at has the true availability of 24/7 personnel trained in airway management is important, and developing contingency plans for unexpected emergent clinical situations is strongly recommended. The second point that the guidance document discusses around phase 1 is stopping criteria. And I've had a significant amount of experience going back and forth with the agency on stopping criteria. It seems to me now that especially for FIH studies, the agency is very concerned that a tailored approach is taken to each drug, which has pre-specified clinical and kinetic criteria of when dose escalation will be halted. They don't want you to use the National Cancer Institute common terminology, and they specifically say that in the guidance document, and we have put that forth in front of the agency previously, and they promptly rejected it, which again, their rejection and then subsequent teleconference and conversation takes time. And so a good learning point is you can't do that. You need pre-specified clinical criteria. Depending on what your drug is, you have to say, okay, if we see this many adverse events of this type, we will pause the criteria, unblind, take a look, and then continue or not continue, et cetera, et cetera. So that needs to be a detailed plan. They want to see that prior to commencing FIH. Okay, and the last slide, a brief word about phase 2 studies, and then we can get to the fun part, which is the Q&A section. And let me first encourage people to write questions and stimulate debate, because I'm very interested in what everyone has to say and what they think about the various points that I've made. And I'd like to hear competing points of view. So for phase 2 studies, the general point that I wanted to make is that the guidance document essentially lays out what regulators want to see in phase 2 studies. And the funny thing is that their way of thinking about phase 2 is often very different than what a small pharmaceutical company especially will think when they envision phase 2. And I state this by basically saying that phase 2 studies serve two masters. From a commercial standpoint, if you're a small pharmaceutical company, you want to show that your drug has efficacy, you want to do that quickly, and you want to do that in a way that is not very expensive. Why? Because you only have a certain amount of time and money, and you need more investment dollars, and you can only get those more investment dollars when you've proved that the drug works. Now, the guidance document shows the regulatory interest, which basically what they want to see is, yes, they want to see that the drug works in phase 2, but they want you to characterize how it works. In what models does it work? And just as important to them, although perhaps not as important from a commercial standpoint, is they want to see where does it not work. So some failed phase 2 programs would be perfectly acceptable to them. And it would actually be helpful because then they would know that, okay, this is a drug for X and not for Y. They also spend some time in the document discussing the fact that they want to know the pharmacodynamic properties of the drug, onset, offset, ceiling effect, a dose that's so low that it's ineffective. These types of things, which in acute pain is much easier to gather because the dental method has the 2 stopwatch method, et cetera, than it is in chronic pain. Okay, rounding the corner here then. In conclusion, I really like this guidance document. It takes us a long way. The FDA took pen to paper and put down issues they've been discussing for years and years, and for that, I think we should all be very appreciative. It provides a reliable framework. I try to think of it as a scaffolding. Yes, there is a lot of regulatory speak and disclaimers, but to the defense of the agency, there really has to be, based on the complexity of the topic that they're discussing. So my advice based on this whole talk is use your regulatory interactions to clarify the what ifs, the it depends portion of your application. There always are going to be those portions. There's nothing that can be done about that, and that is really the purpose of your regulatory interactions. Again, I appreciate everybody listening to this webinar. I'm looking forward to answering questions.